Thanks everyone for joining today. This week's seminar speaker is Dezene Huber, joining us from the University of Northern British Columbia. Dezene has been working on bark beetles since the 90s and did his bachelor's degree in the University of Calgary, PhD at Simon Fraser University, postdoc at Davis. And he's now, as I said, a full professor at the University of Northern British Columbia, Faculty of Environment. And a fun fact about Dezene, he taught me molecular and cellular physiology. So it's fun to have him here today and see what he's been working on since I had him as an undergrad prof. So Dezene, I'll let you take it over from here.
Yeah, and thank you so much for inviting me, Brock, and to your entire department. And I'm sort of sad I can't be there in person, and maybe someday I'll be able to come by. And yeah, having Brock in my class, and then this is quite a while ago. He said, "Don't say it was a long time ago," but it was quite a while ago now. And then seeing him here is great. It's good to see our alumni doing their thing around the world. So I'm going to, as you can tell from the title, I'll be talking about bark beetles and mainly about mountain pine. Well, I think entirely about mountain pine beetle. In fact, I'm just going to share my screen here and see if I can make in Zoom world here. I find I need to ask if, can you see that I'm moving a laser pointer around the screen here?
Sure can.
Perfect. Okay. So this talk is untested anywhere, so I'm hoping I don't take it too long. I'm going to try not to. I don't have a clock in front of me anywhere, so if I start to get towards too long, if Brock, if you'll type in, I have ways of skipping past a few things here. But yeah, as Brock said, as Brock said, I am from the University of Northern British Columbia. I'm a professor there, and our graduate program, I've just decided I'm going to advertise our graduate program a little bit. We're always happy to have new graduate students here. This is a view of our lovely campus. It's a really, really beautiful part of the country. BC, of course, is really beautiful, and our part right in central BC has a lot of great features to it. We're really close to the Rockies, lots of good hiking and fishing, and just a really good small university to be at. Lots of really personal attention. Our graduate program, you can see the link there on the left if you're interested in finding out more about it. Graduates quite a few grads a year, and they all end up in fantastic jobs related to what they have been studying most of the time. So we're quite proud of our small but strong and powerful institution in terms of research and teaching and other aspects, service to the community as well. A great deal of really local research, sometimes quite hyper local. I'm starting with a acknowledgement slide here because I like to do that. This project, the research I'm discussing here is part of what we call the TRIA project, and it went on for about 10 years, from about 2010 to 2020. No, I'm a little bit wrong on that. 2008 to 2018. I'm trying to remember my exact dates. But we had a ton of people worked on it in different aspects over the years. And most of my slides will be a little citation at the bottom, and most of the time, not always, but most of the time, those are various TRIA researchers, sometimes associated with me. But there are a lot of others as well. Yorick Bowman and Janice Cook co-led various portions of this project, and we had a lot of really great project managers as well over the years, and then just too many people to acknowledge here. Dozens and dozens graduated many students out of this project as well, from University of Alberta, UBC, UNBC, and various other universities as well. We were funded by mainly Genome Canada, Genome BC, Genome Alberta, and NSERC, and some from Alberta Innovates as well, plus another set of other contributors in different ways. So the mountain pine beetle, probably many people have heard about this little critter. Part of that is because it's so well studied now, but in terms of a very basic understanding of its life cycle, the adults emerge from host pines here right at the beginning of summer, usually around the second week of July or so, and they disperse off to new hosts after they leave their host tree. They live in just underneath the bark in the cambium, and they mine around in the foam as larvae, and then they finish their development here and disperse. And they aggregate using aggregation pheromones, and they reproduce in the new host tree, and then the larvae develop. And they develop over the course of the rest of the summer and the warm part of the autumn. But at some point, they begin to develop overwintering compounds and other overwintering physiology as well. So that it'll take them through the winter. And they can survive some pretty cold temperatures near to -40 in some parts of their range. And then in the spring, they come back out of overwintering, so they have to adjust their physiology again, and they finish their development, and then the new generation emerges. And in this talk, I'll be talking about overwintering, definitely, and hopefully a little bit about some of this host colonization and reproduction as well, in terms of the genomic work that we've done. But one of the most important things about these beetles is that in order for the adults and the larvae to survive, the adults need to, once they attack the tree, they need to kill the tree, because the tree is going to do its best to kill these invading beetles as well with its various secondary metabolites that are contained in the resin. And so the attacking, and this is probably actually, I've tried to find a good Dendroctonus. This is most likely, it looks to me like a spruce beetle or a Douglas fir beetle, but we can pretend it's a mountain pine beetle. But either way, most of the Dendroctonus, the genus Dendroctonus in which mountain pine beetle resides, do the same thing. They attack and aggregate on trees. And if only one or very few beetles attack a tree, which is typically the case, there's not a ton of beetles around in non-epidemic situations, that beetle will probably die and the tree will win. And this is a good example of a beetle that lost its battle with the tree. It begins to dig through this outer bark, and it starts to sever resin ducts as it gets just underneath the bark. And the resin ducts spew out this sticky resin that's full of mono and sesquiterpenoids and diterpene resin acids and other nasty things as well onto the beetle. The beetles are quite well adapted to swim around in it. They're able to sort of, not hold their breath per se, but limit their respiration to some extent. And they also have a lot of detoxification enzymes like cytochromes P450 and glutathionase transferases. But oftentimes the tree wins if it's able to mount enough of a defense against these insects. But if a whole bunch of beetles will attack the tree at the same time, then the beetles and their fungi, which they carry in little pockets on their bodies called mycangia, the fungi start to grow through the tree, and the beetles sever enough resin ducts, and the tree starts to expend all of its defenses. Then the beetles are able to overcome the tree's defense, and they win in that case. And this is a successful mountain pine beetle attack on a lodgepole pine, and each of these little yellow dots is the entrance tunnel for one pair of bark beetles, a male and a female. And this is an amazing behavior if you've never witnessed it. We know what the pheromone components are. We can place those onto a tree, and in a really heavy infestation like we had here in most of the interior of BC about 10 or so years ago, you could literally sit there and watch it happen on trees and have the beetles landing all over you too. I had a few beetles try to bore into my neck at various times where the infestation got to be so heavy. But this is all coordinated by aggregation, and then later on by anti-aggregation pheromones once the tree fills up to tell incoming beetles that this tree has no more vacancy. And also the chiromones, so the terpenoid, mainly smells that are coming out of the tree as well, commingling with various pheromone components that the beetles make or convert from some of the tree compounds. And really, so in this talk, I'm just going to be talking about mountain pine beetles. So right down here, this vector. But in a lot of ways, this is the entire TRIA project. We're called TRIA because this is really a disease system of really at least 3 genomes. The tree itself, which is in the case of mountain pine beetle, which is typically a pine. We know that they can attack spruce in some cases, but typically a pine. Most pines in North America that are in the same Chira range or Sympatric and mountain pine beetle are susceptible to the mountain pine beetle. The one exception that I know of in California is Jeffrey pine, but most every other pine is susceptible to pine beetle. So when we say at least 3 genomes, there's actually several pine genomes involved, 1 pine beetle genome, and then also several fungi that they carry as well. So it's actually more than 3 genomes. I guess it's sort of 3 groups of genomes. But we really need to think of this entire thing as a disease triangle with a vector, a pathogen, and a host. And that's similar to a lot of other disease triangles out there, including things like malaria, for instance. And so we've always sort of felt that our work has some parallels in a lot of other work in agriculture, forestry, and even medical entomology in a lot of ways, in terms of at least analogies and models that could be followed for understanding systems. This is a picture of the infestation in British Columbia back around 05 or 08, very shortly after I started at UNBC, when I was able to take a helicopter flight with one of my colleagues, Dr. Staffan Lindgren, to over some of the area near to Prince George, BC, so right in the very center of British Columbia. But all of these trees here are pines that have been killed by pine beetles in the year before. And so within about a year or so, the needles become quite chlorotic and then red by sort of late spring, early summer, and they fall off the tree. So by the time you see a red tree, that tree's been under attack and has been dead for about a year. These other trees in here, I'm not sure what those are. They're either pines that haven't been attacked yet, or they're things like spruce or Douglas fir or other things that are not susceptible to attack. But you can also see, well, yeah, I guess these are probably trees that have been killed about 2 or more years ago, or the needles have all fallen off. And so this is the type of landscape that we encountered in central BC right about the time I started my my research chair at UNBC, and for the next several years as well. And this landscape is moving across the province and into other parts of Canada as well. In BC alone, though, about 700 million cubic meters of pine timber have been killed, regular harvestable economic timber. That's affected in total about 18.3 million hectares in BC, just BC all by itself. And that's 20% of BC's land area. And that's about, if for that size is about twice the size of Indiana. In fact, I looked up these numbers. I didn't know, that means BC is, I guess, 8 times the size of Indiana. It's a very huge place. But I mean, put 2 Indianas together and that's how much pine space, pine land base has been affected. This is from a paper not related to TRIA. The reference is down there. Corbett et al. Said there will be a cumulative present value loss of $57 billion in GDP and a 90 billion decline in welfare, which is a compensating variation, which is an economic term I don't understand, from 2009 to 2054 in BC. So large, I mean, immediately close to $60 billion down the drain if you look at timber as simply a harvestable entity, and that's it. But I mean, long-term GDP impacts as well. And of course, this has had massive effects on families, thousands of families across BC. Brock, from being from this area, would know about this as well. I mean, most of these towns here have mills, and much of the, especially the smaller centers around about Prince George and other parts around Kamloops and Kelowna, really tend to be reliant on the forest industry, either directly or indirectly. And then beyond that, there have been short and long-term effects on the landscape and ecology. Of course, this insect is a native insect, but this type of outbreak is not a normal type of occurrence. And I'm not talking about it in this talk, but there's plenty of discussion as to why things got to be this case. And it probably boils down to things like climate change, which is human caused, of course, plus silvicultural practices, and maybe some sheer bad luck as well, intermixed to be a perfect storm. This is a map of Canada. I'll show you a map later, which has a little bit more range shown, but the dark to light gray shows the pine volume from low to high, dark being high, light being low across the country. Most of the pine in this area in BC, which is very pine heavy, is lodgepole, plus some ponderosa pine and white bark pine down in the south, and so at some of the higher elevations. But once you start moving into this area, you get to jack pine and red pine and other types of pine as well. This is the amount of, this is the cumulative mountain pine beetle. This is from Cullingham et al. 2011, who were part of the TRIA project. This is a cumulative mountain pine beetle attack over, about 60 years or so, no, 40 years or so, from the mid 1900s to close to the turn of the century across British Columbia. You can see that it's basically, in terms of just BC, it's basically limited to BC with some small little incursions into southern Alberta here. 99 to 2005 is when it really took off. And then we get into 2009 here. What you see here is lodgepole pine range, and this lighter color here is jack pine range, and you can't see it very well, but there's a pocket right in here of lodgepole and jack pine hybrids. So about 2009 or so, the beetles moved into these, they moved across the Rockies up in the north. So Prince George is actually sitting right about there where my laser pointer is. The beetles moved across the Rockies into sort of north central Western Alberta, into this, started using hybrid pines, and then started using some of these jack pines right in this little area here. And so now we have pine beetles in lodgepole pine and using jack pine for the first time in a brand new geographical region. This is a different picture of the same situation. So that's the infestation right there blowing up in that spot in British Columbia. And this is lodgepole pine. This is jack pine, which extends down here, not quite to Indiana, but close, through the Great Lakes region. And so that's the amount of space that mountain pine beetle has now opened up for itself to move across Canada into parts of the US. Plus there's other pines, like I said, red pine and others in this area as well. Southern pine beetle down here has been making incursions going north. At some point, these two species are going to meet again. I heard entomologists at one point say that they meet again for the first time in eons, being sister species. So I have no doubt that that will happen, possibly within all of our lifetimes. This paper, not from TRIA, but was right at the very end of TRIA. It was published by Windmiller Campion. They wrote, there are still many unknowns regarding mountain pine beetle in the Great Lakes region, so your region. However, as natural resource managers plan for the future, they may want to consider the potential arrival of mountain pine beetle in eastern pine forests when developing silvicultural prescriptions. So people are indeed thinking about this already and this possible occurrence. And so a lot of our work here in BC and Alberta is hoping to understand what's going on with this movement. And a lot of that relates to overwintering and also host colonization, because as the beetles move into colder areas and also new hosts, understanding how they're dealing with the cold and then the new hosts is going to become more important. Early on in the TRIA project, we developed a transcriptome of a mountain pine beetle transcriptome. This is the paper here on the right. We derived it from 14 different libraries from a variety of life stages and tissues. So larval, early and late larvae, pupae, antennae, gut tissue, mid-gut tissue, where they produce their pheromone, hormone-treated insects, overwintering insects, to give us a really good diversity of transcripts to sequence. And early on, that transcript is really useful for some of the initial functional genomics work, which I'll describe here, and then later on annotating the draft and then the full genome. Shortly after the transcriptome, we published the first draft genome of the pine beetle about 9 years ago now. And we really focused in, once we had gotten that genome put together in this paper, on cytochromes P450 and glutathione S transferases, which are often involved in detoxification of host secondary metabolites. And also one of the really cool and interesting things that we found in this paper was that there is at least one horizontal gene transfer of a bacterial sucrose 6-phosphate hydrolase. What that's doing there, we don't know. Prior to us, a group working on the coffee berry borer, which is also a bark beetle, a skeletine, had found several horizontal gene transfer events. I think a mannose, something related to mannose metabolism, for instance. And since then, a lot of people have been finding we know that horizontal gene transfer concepts have really taken off rapidly across the ecological world and our understanding of how things interact. But in some bark beetles, there are large numbers of horizontal gene transfers, and it makes some sense because they interact with so many other organisms during their life cycle that some of these events could have occurred in the past and then been favorable in terms of something related to their metabolism. So that was one of the cool things from this draft genome that I always like to mention. And so take a look at that if you're interested. Very recently, in fact, in the past few months, we've published a very high quality genome for the mountain pine beetle. It's a chromosome level assembly, and I'll describe it in a moment in some detail of the mountain pine beetle. The N50 values, which is a measure of the quality of the genome in terms of scaffold size, so the size of the pieces that we're putting together, were improved by somewhere in the range of 30 times compared to the draft genome. And the gaps between the scaffolds were also really substantially reduced. And as I said, it's a chromosome level assembly, and we ended up recovering 12 large scaffolds from each male and female genome, and they were very much parallel scaffolds, which helps us to believe that what we found were the 12 chromosomes of this insect. 11 of them are autosomal chromosomes, and then one is what's called a neo-X chromosome, and I'll describe that thing in a moment as well. But this figure here on the right, which is from the paper, shows there's centimorgans are shown on the scale on the left, and then on the right is a recombination density in various regions of these what we think are the chromosomes of this insect. We numbered them across this way, as you can see, but there's some, depending, the two sizes here got mixed. The scaffolds here are not, there's something related to this is in centimorgans, not in actual size, and these are numbered in actual size. So 10 and 11 here, the scaffolds, they're pretty close to the same size, and the scaffolds sort of mixed up. So what 10 is and what 11 is a little bit up in the air in terms of size. But we were able to assemble these really nicely, and you can see there's some areas of where there's a lot of recombination going on, and especially right here in chromosome number 4, and I'll get to that here in a second as well. So there's a really interesting sex chromosome system in mountain pine beetle. They have what's called a neo-X, neo-Y system, where the neo-X chromosome is a fusion of an ancestral autosomal region plus the ancestral X chromosome. And so it's the fusion system. And so there's neo-X, where you have the X, the former X, plus this autosomal chromosome. And so this other half of this pair without the X added onto it becomes the neo-Y. So this thing here used to be an autosomal chromosome, but now it's become a sex chromosome. We didn't recover the neo-Y chromosome, and that there's a number of technical reasons for that, but it's really hard to differentiate from the neo-X. But we did find a bunch of, so there's some known neo-Y scaffolds that really mapped quite well to the male, what we believe is the male neo-X chromosome. And then there's a whole area where nothing mapped at all to it. And so that seems to be the ancestral X region. So we think we have a pretty close grasp of what's going on in terms of the neo-X and neo-Y chromosome, but there's still more to be found in that area as well. But there's some complexities related to sequencing in that area because of similarities between the two. We looked very closely at each of the chromosomes, and this, you can see, this is chromosome 1, or this neo-X chromosome. And it goes all the way down here to chromosome 12. You can see that right there. And this shows FST, in which we are comparing north and south populations, so southern BC populations with some of the northern populations, which were the ones that introgressed into Alberta. And there's a fair amount of variation in some regions, which tells us that which shows us this variation between north and south populations. So there seems to be some active evolution going on there between these between these two populations. And interestingly, using a measure called Tajimas D, which helps to find indicators of non-random selection processes, there are two areas. So one at this very end of the neo-X chromosome, and then one sort of near one end, but in the middle of this chromosome number 4, that showed really elevated regions of elevated levels of this Tajimas D indicator. And if we take this part right here and just zoom in on it, that's what you see here. And you can see the two different, so I think red, if I try to remember here, red was the northern population and blue was the southern population. You can start to see some divergence there in this Tajimas D measure. And you can see a great deal of it here in this chromosome 4. And it turns out that chromosome 4, we tested it every which way, and it seems like this is an entire inversion of this chromosome in the northern population. I'm trying to remember which population. I think it was the northern population, but there's an inverted region, it seems. And the genes that are in this particular region of chromosome 4 is really enriched in genes related to gene regulation and also to protein synthesis. So something's going on there in these two regions, particularly in this region of chromosome 4. We're not sure what it is, but the high quality genome helped us to understand that something is going on there. There's certainly a lot to be done now that we have that information. You can see this very high region of FST also there. So using all those different genomic resources in different ways, we studied overwintering, and very early on in my lab, so going back to one of my first graduate students, Jordy Fraser, and then Tiffany Bonnet, who was an undergrad who worked with me as well. We started to look very closely at glycerol biosynthesis, because even before we had large-scale transcriptomics data, we knew that mountain pine beetle started to develop a lot of glycerol in its system as the winter hit, but we wanted to see which genes were being transcribed, and if we could see a pattern across the autumn and then across the spring. And the graphs here on the right show qPCR results for autumn mountain pine beetle ranging from September until November. So 5 different time points from September until November using qPCR. And up here, these lines show the temperatures. So this is on the left is a fold change in transcripts, and on the right is a temperature, the average temperature in degrees Celsius with minimum, maximum, and mean. And we found a whole bunch of transcripts related to glycerol production that were upregulated in the autumn. So you can see in these, this is a multiple comparison test that's being done here, but you can see that these are upregulated across the autumn significantly. And we found out that the beetle transcripts related to lipolysis did not show any significant change. So it seems that the beetles are making glycerol, from glycogen, so stored glycogen, and also gluconeogenesis as well. And then we can see so we see this ramp up of various transcripts in the fall, and then in the spring, those same exact transcripts. So it's the same type of graph here with the temperature and then the fold changes. These same transcripts, and the 6 graphs are arranged the same way, show significant decreases quite quickly in the spring as well. So what the beetles are doing there is metabolizing the glycerol, and they're using it, these are larvae at this point, and they're using it for development, we believe, but it's not converted back into glycogen. It's probably really rapidly consumed as temperatures warm and as the beetles, as the larvae need to develop. So it's not converted all the way back to glycerol from the way these transcripts look. So very shortly after that work, which was very targeted on one metabolite that we knew was important in this insect for overwintering, we started doing large-scale transcriptomics and proteomics work here. I'm going to show you just the transcriptomics work here, but the proteomics, this is a proteomics paper referenced down here because this figure comes from it. And it shows the sampling times again with temperatures across the season from September until November. It's the same sampling times as previously with that glycerol study. In this case, we took 4 because of the amount of data and also the cost at that point. We took an early and a late fall, and then an early and a late spring sample in each case for both the transcriptomics and the proteomics work. So we're comparing apples to oranges. As I said, I'm not going to talk about the proteomics in any detail here. I have some slides at the very, very end if anyone really is interested in looking at those. But the transcriptomics and the proteomics really matched each other in terms of what was going on and really verified a lot of what was going on with overwintering in this insect. So we sequenced for transcriptomics n equals 4 in each case, the transcriptomes of September and November in the autumn, and then March and May larvae in the spring. And we found 581 transcripts that showed differential transcription in autumn, 534 that showed differential transcription across the spring, and 145 that showed differential transcription in both seasons. And of those 145, the ones that we really found interesting were the ones that would ramp up, the 107 that showed a pattern of ramping up in the autumn and ramping down in the spring, or the 29 that ramped down in the autumn and then ramped down in the spring. And so we really focused in on that particular bundle. So even just a subset of this 145, a large subset of that 145, but a subset nonetheless. One of the things that we found in this transcriptomic work was that that glycerol qPCR work that we had done previously was really well verified by this large scale transcriptomic work. And among all of those results from the qPCR were verified in this large scale transcriptomic work. And one of the interesting things about the glycerol pathway, so if you have glycolysis up here, glycogen storage, glycogen conversion, you get right near to the end of the pathway where glycerol is being produced. So it can be produced via lipolysis, and we've already ruled that out, and the transcriptomic, large-scale transcriptomic work also ruled that out for glycerol production. But you have two potential routes to get to glycerol, either through alcohol dehydrogenase or through glycerol kinase, or well, I guess G3, glycerol 3-phosphate dehydrogenase and glycerol kinase in that order. And Jordy in his qPCR work had determined that it seemed to be coming down this branch of the metabolic pathway and not this branch. And the transcriptomic work showed that as well. So these two particular, the transcripts for these two enzyme products showed increases in the fall and decreases in the spring, with sort of almost stasis in the middle of winter, sort of comparing between November and March. So it seems that this pathway, as Jordy had thought, is the one that is actually working in this insect to produce glycerol. We saw a lot of other interesting things too, and I can't get into all of it. The paper is referenced down at the bottom here. But we saw, for instance, a lot of chaperones, heat shock proteins ramping up in the fall and ramping down in the spring. And this tells us that there's a lot of cellular and physiological stress going on that the insect needs to manage as it prepares for overwintering and as the temperatures start to get worse as well. It's also dealing with, as we found out later, very high levels of terpenoids, because even though the adults have killed the tree has changed the terpene content and the toxicity of its resin. And even though the tree is dead, a lot of these toxins remain in the tissues. So the little larvae are not just dealing with oncoming cold, but also an extremely toxic environment. So some of this chaperone heat shock protein type of situation really makes sense in that context. As I said, the proteomics, oops, where are we at here? Okay, 10 minutes to go. I think I'm good. We'll probably not talk about host colonization, and maybe a little bit, we'll see. In terms of overwintering as well, we as I said, the proteomics work really verified a lot of what we saw in the transcriptomics. And then we started to look at the metabolites specifically, because we knew that there were, from some of the transcriptomic work, we knew that it was more than just glycerol. We saw indications of other things. For instance, we saw trehalose as well. This work is with a former postdoc of mine, Philip Batista, who's now with the BC Ministry of Forests. He looked at, along with Kathy Bleicher, who's a Canadian Forest Service scientist in Victoria, he looked at glycerol concentration in BC beetles, BC being, when he says BC, this is Victoria, which is on Vancouver Island, which is very warm. It's sort of compared to much of BC, it's sort of Seattle-like or even warmer situations. And Alberta beetles near Whitecourt, which is northwest of Alberta, which is very cold. And so glycerol concentration in overwintering beetles populations differs. We put them in the same cold situation. So it's sort of a provenance trial. And the Alberta beetles ramped up their glycerol concentration massively, whereas these Vancouver Island beetles did not at all. The Vancouver Island beetles maintained high levels of trehalose, whereas the Alberta beetles did not. And also the supercooling points differed significantly as well, with the Alberta beetles developing very deep supercooling points and these Vancouver Island beetles not at all. We also looked at them deacclimatizing, so coming back out into a simulated spring. And at that point, they become much more similar again. So the Alberta beetles drop their glycerol concentration down to close to what the BC beetles are at. And the Alberta beetles start to increase. Remember, they had like very floor level trehalose levels. They start to increase their trehalose as well again. So this tells us that what we saw in the proteomics and transcriptomics work in one population of beetles is probably valid for that population, but some of the things may be valid for all populations of mountain pine beetle entering into overwintering, but there's also going to be some differences as well, depending on their population. And mountain pine beetle ranges from Mexico around the Great Basin through Canada and down to the Black Hills of North Dakota. So we expect to see a lot of differences if we start to look more closely. We also were fortuitous more recently in this paper down here, which we published a couple years ago now. Kirsten Thompson, who's a PhD student, who's just finishing up now in the next few months here at UNBC, and Brett Murray and myself worked on adult mountain pine beetles. So we're overwintering right on the border of British Columbia and Alberta, just inside of BC. This is the border along the continental divide here. Normally, adults sometimes will make it through the winter, and some reports from further east here getting into Alberta is that there's a lot, in some cases, the beetles are not completing their larval life cycle in their summer. They're late as they come into summer of their larval life, and they end up being 2-year cycle bark beetles, which then emerge. And so the adults then have to overwinter instead of the larvae. And so we thought that was pretty interesting. We found one of these populations at a little provincial campsite that was being killed by pine beetle, and sampled across the entire season until a really cold snap killed all of the beetles at that point when things get close to minus 35. But we did metabolite profiling of these insects, and we found a whole bunch of different compounds that showed shifts across the study period. 3 of those were glycerol and trehalose that we already knew about, and also proline, which we had suspected from other research, other transcriptomic work, plus a bunch of other things. Most of these, it turns out, are amino acids of various types. But we focused in on these 3 because they're all known cryoprotectants, and they all have some pretty interesting characteristics as well. It turns out the glycerol levels in these adult mountain pine beetles, so this is mean temperature here, increased over the autumn, as we've seen before. So that wasn't overly surprising. Trehalose increases and then decreases. So there seems to be a bit of dynamics across the autumn in these adult mountain pine beetle. And proline also increases over the cooling period in the autumn as well. Trehalose, we know, plays a number of roles as an antifreeze, including stabilizing proteins and cell membranes, and it's also a storage carbohydrate in a lot of insects. Proline is interesting because we know that it has a role in depressing the supercooling point in a lot of insects, but it also has a dual role. Later on, it's used in flight energetics as well. So it's possible that the insect is increasing the proline levels to help it overwinter, but then also using that proline later on when it gets into its dispersal flight. But the these results were interesting because it was adult mountain pine beetle compared to larval mountain pine beetle, and may be related then to the spread of the insect into forests further east as it encounters colder environments. This paper, not from us, tells us that there's really a lot more to this overwintering story yet to be found. These folks looking at Dendroctonus ormandi in China found an antifreeze protein that was active in that Dendroctonus species related to mountain pine beetle. We know from some of our genomic work that there's at least one suspected antifreeze protein in mountain pine beetle as well. We haven't ever seen it shifting in it's in the transcriptomics or proteomics yet, but we are looking hopefully to characterize it at some point to see if it really is an antifreeze protein. But I think there's a lot more to be found even beyond that. I probably only have a few minutes here, so I'm going to just quickly talk about some host colonization work, and I'm going to skip by a bunch of slides, but I'm not going to skip by this bunch. This is work by Victor Shigelski, who finished his PhD about a year ago now, and is working as the curator at the Insect Museum at the University of Alberta. He did his PhD at the University of Alberta with Maya Evenden and Felix Sperling, and I was on the committee as well. And working, pardon me, with Maya, who runs a lot of flight mills, where she has insects flying on flight mills, and then looks at their various things related to their physiology and transcriptomics, etc. He put pine beetles onto flight mills and had them fly, and was able to sort them into two groups: either strong flyers, which spent a lot of time in active flight, or weak or non-flyers, which spent minimal time in active flight. And he found 3,000 genes that show differential expression between these weak and strong flyers. These are insects that would have just emerged as new adults and are looking to colonize a new host. And when he looked across at differences between weak and strong flyers, he found really substantial metabolic upregulation in strong flyers, including lipid metabolism, glycolysis, and things related to the citric acid cycle, which makes sense because they're using so much energy in flight. And some of these insects would fly for kilometers on end on these flight mills. He also found upregulation of muscle-related genes, such as for collagens and myosins as well, so motor proteins and call it structural proteins in the strong flyers compared to the weak or non-flyers. A lot of oxidative stress response genes, DNA repair genes, antioxidants as well, including GSTs in the strong flyers, and also detoxification enzymes, seemingly that the beetles were getting ready to attack a tree and burrow into it and deal with a lot of the host toxins. He showed downregulated nocturin, which is a circadian light response protein in the strong flyers, and the strong flyers interestingly flew a lot more, significantly more during the dark phase than the weak flyers. So it seems that strong flyers seem to be somewhat predisposed to flying at night, related to their circadian rhythms, at least in terms of this particular protein. And also their olfactory associated transcripts were downregulated in strong flyers as well. And we're not sure why that's going on, but one possibility is that they remain unresponsive to host compounds and pheromones for some early part of their flight, so they don't just land on the nearest tree. Instead, these strong flyers seem to have some sort of adaptation that causes them to fly by trees that might be being colonized and head even further downfield. And so we end up with these two populations, sort of weak flyers that spill over into the nearby trees, and then stronger flyers that go a far greater distance and spread the infestation further. So this, of course, relates as well to a lot of this dispersal capability, especially in the new range. And I'm going to end here with this slide related to Victor's work as well. There were 3 SNPs that we found as well, 2 of them in exons of functional genes, and 1 of them in an intron that showed that were associated very strongly with flight distance. So the 2 exons were associated with flight distance, the strong flyers having the two exonic SNPs were associated with flight distance, the strong flyers having these two SNPs, and the propensity to fly, so the likelihood that they'd keep flying if they stopped, they'd start again, was related to this intronic SNP. We're not really sure how these are affecting flight. This is one of these areas that still needs a lot of research. For instance, one of these exonic SNPs is a conservative mutation. It's not changing the amino acid in the resulting protein. And of course, this other one here is in an intron. So if any of these have any functional relationship is yet to be seen. I won't go through, we did some transcriptomics work as well on host colonizing pine beetles. We saw things like cytochromes P450 being increased and decreased as insects fed on trees. I'm not going to spend any time here because we probably should get to an end, but we also saw chaperones ramping up as the insects would attack their new host, related probably to physiological stress. And we saw that the females and Victor saw this as well in his flight work, their vitellogenin levels were very low, or the transcripts for vitellogenin production were very low in dispersing beetles and in beetles that had not yet fed on a host. But once they fed on the host, the females' vitellogenin levels ramped up. So probably they are, the females are saving all of their energy for flight dispersal and for the initial attack on the host. But once they've gotten into the host and have overcome its defenses, they begin to take all of the rest of the energy that they have and start to produce vitellogenin as they produce eggs to provision their young. So it's, in a lot of ways, it's like salmon, if we're talking in terms of BC analogies here, using as much as using the energy that they need to get to their spawning grounds. But once they get there, they use everything in their body at that point to ramp into production to produce the best possibly provisioned larvae. And so that it seems to be that pine beetles and probably many other dendroctonus are doing that same thing. So to summarize, we've developed high quality genomic data, including a chromosome level assembly for the mountain pine beetle. There's a bunch of interesting features, which I think we've only barely begun to explore, including horizontal gene transfer and various signals of selection as the insect is moving into new regions of Alberta, for instance. We've used genomic data as a foundation for understanding host colonization and overwintering of larvae in 2-year cycle adults. And these 2-year cycle adults may become important in this new colder geographic range. And overwintering in adult beetles shows substantial differential gene expression and metabolite accumulation. So for instance, things like glycerol, trehalose and proline metabolites, and a lot of their transcripts as well, and also things related to cellular and physiological stress. Dispersing and host colonizing beetles showed differential regulation of detoxification systems and in females vitellogenin production related to their attack dynamics on the host. And now we're focusing our attention in a similar, maybe not quite as extensive way in terms of the number of people involved, but on spruce beetles and some other related bark beetles as well. And I'll just finish up with that slide of acknowledgements, and I guess it's time for questions. I hope I haven't gone too far over time. Thank you very much.
Thanks, Dezene. Thanks, that was fun to see. So if anyone needs to log off, of course, you are welcome to. We've got time for, well, we'll see how many questions we get, but probably time for 1 or 2. Use the little hand raise icon just so I can see you, and you're welcome to just ask it outright once I acknowledge you. Give folks a moment. I definitely have one. So I'm actually curious. You were talking about glycerol production, the importance of antifreeze compounds to help mountain pine beetle overwinter. I'm curious. Did any of those genes that you identified associated with the production of antifreeze proteins, did those overlap with your high FST regions between north and south?
There is. So there's one, actually, Brent Murray, who's a prof here at UNBC, working with Linda Horianopoulos, who's was an undergrad student, is now doing a PhD, I think, at UBC. They found an inhibitor of apoptosis, interestingly, related to cold tolerance, that showed some selection, so it showed population differentiation between north and south. I'm trying to recall her research exactly, but I think she did some characterization of that as well. And that seems to be related as well to cold tolerance as well. So oftentimes you end up with a fair amount of potential, I think, cell death and other things going on. So there needs to be some regulation for apoptosis. In terms of a lot of these other things, we haven't seen that with some of the population genomics work that we've done. We have seen, and so Phil Batista, whose work that was at Vancouver Island versus Whitecourt work, has been finding some population differentiation in some of the P450s and glutathione transferases. So there are definitely some things going on in that way as well, and that would possibly relate to host use in that case.
Okay. Yeah, I have a few more on that angle, but I'll let someone else jump in here for one more. Oh, yeah, go ahead, Niels.
I had to. Can you hear me?
Yep. Yes, they can.
Oh, all right. I had a question about the anti-aggregate hormones. I don't know if you or others have looked into this, but would it be possible to inoculate trees with these pheromones or a mock pheromone, so to speak?
Yeah, I mean, so I mean, I think there might be some possibility of doing that type of thing. So the anti-aggregation pheromone, the beetles turn it on, quote unquote, when the tree starts to fill up in competition, when the tree is dying, and they're basically saying no vacancy. And so, and they're quite powerful. I've used them in some of my previous work where we were trying to protect trees from insects using combinations of non-host volatiles and anti-aggregation pheromones. This one in particular is called verbenone, and also there's frontalin, which in high levels is an anti-aggregant. I think, it might be possible to do that. The reality, though, is we can make these things in industrial levels and put them into basically what look like almost little air fresheners, basically, and just staple them onto trees. So much more low tech way of doing it, but a lot easier. And a lot other problems with potentially doing that too are that so I mean, forestry and agriculture. A lot of modern forestry tends to look a little bit like agriculture, but it still is a natural landscape, and so there's a lot of reticence, and I think understandably, about genetically modifying trees in various ways that then would spread that gene further, and who knows what the other effects are on other creatures. So because a lot of beyond pine beetle, a lot of insects respond to these beetle pheromones in parts of their life cycle too, so you can imagine some ripple effects in the ecosystem beyond.
If you were to implement such a system, then it would of course be better to have a transient. It will be gone if you leave it as it is, just in case something goes wrong with it. So and I mean, the just stapling little air fresheners onto trees is really pretty tried and true, basically, and pretty easy. But we've there have been things we've talked about before too, and maybe making trees that produce various things to inhibit bark beetle, like basically putting pharmaceuticals into the trees to inhibit their production of antifreeze compounds or other things like that. In that case, you could theoretically treat trees and just fill them up with whatever, or make them make this pharmaceutical. The trouble otherwise too, when you saw that landscape as well, like treating large areas like that, it becomes very difficult also. And then you're talking about decades as well between rotations, so like 50 to 70 years as well, instead of 1 year agricultural crops too. So it really, there's a lot of things that make some of dealing with this insect difficult. Good questions, I think.
Yeah, thanks.
One more question before I have to go. I have a question about the fusion of the Neo X chromosome. Was it an end-to-end fusion, or was it part of the original autosome lost in the fusion event?
I think there, I'm trying to recall this now. I think there was there's a little bit of evidence of some loss. There's been some karyotyping work done with other dendroctonus as well. Not all of them have this system, so I think that is determinable. I think there has maybe been a little bit of loss, but I think it's mainly a straight-on fusion, as far as I recall. There's yeah. I'd have to look a little bit more closely into that. That's a good question. It's sort of an odd little system, actually, and it certainly does make the sequencing a little bit more difficult.
Thank you for the interesting lecture. And
Yeah,
also
thank you
your
for coming.
answers.
Yeah, thank you.
All right, thanks everyone. We're going to end it there. I imagine Eric has to go set up for another defense, and we're a little over time. So I dropped a little line in the chat there, thanking you all for coming and reminding the postdocs and grad students to join on with the lunch for Dezene. Bring your questions. Lunch, I guess breakfast for you, right?
Yeah, I haven't had breakfast yet.
Perfect.
Yeah.
So yeah,
Great.
thanks everyone, and thank you, Dezene.
Yeah,
That was awesome.
Thank you. Thank you.